then the most recent conference I went to, you started seeing lots of people talking again about knowledge graphs and graph-based approaches, and even how to combine that with things like large language models. Hi there, welcome to episode 1 of Peers Reviewing, a DrugBank podcast where we dive deep into compelling research in the biomedical space. My name is Chris Klinger, and I'm the scientific lead here at DrugBank.
And I am Marisol Garcia Patino, scientific support representative here at DrugBank.
We spend a lot of time reviewing emerging research, and we picked one particular paper that has sparked our interest to share with you today. So in this episode, we will discuss the paper, Network Expansion of Genetic Associations Defines a Pleiotropy Map of Human Cell Biology, which was published in Nature Genetics in March of 2023. And of course, we'll share the link to this paper in the description for your reading pleasure. Okay, so let's get right into it. So just to give everyone a sort of brief idea of what happened in this paper, of course, if you end up reading it later, you'll be able to expand on this quite substantially. But the basic idea here was that they built a very large biological network centered around protein-protein interactions. And then they sort of used genome-wide association study related genes as seeds into this network to perform what's called network expansion. And from there, they were able to build out these sort of modules, as they call them, of proteins that are, conceivably important for a given trait, and did a bunch of sort of work and analysis around that. And what I find interesting about this is that, GWAS data is very useful, but it can be difficult to interpret on its own. These issues around associated loci not directly corresponding to an actual gene. So you have these intragenic regions that show up that then need to find, okay, which gene is this actually associating with and why is it important for the condition under study? And the network stuff is just a really nice way of sort of tying a bunch of different related pieces of data together. That was what I was sort of thinking about it. What did you think overall about the paper, Marisol?
Yeah, I think that this paper just represents the next step that people are doing when it comes to GWAS studies. So back a few years back, you could see that there was a lot of publications of GWAS studies done, but a lot of those studies just stopped there. And there were some discussions on how a lot of those variants that were being identified, they were not linked to any causal genes. Like they were there, but sometimes they fell in areas that are not being expressed or that were really hard to link to a specific trait. So this type of approach is really, really helpful just because it gives you the extra information that a single GWAS study without any of these other components would give you.
Yeah, definitely. So I think in terms of really understanding this paper, because it's quite dense, as you might expect for a paper published in Nature Genetics in this day and age. But for my mind, I think that 2 of the 5 figures, so figure 1 and figure 3, to me, are the really important ones to understand for anybody who wants to get a sense of what this paper is all about. Did you find the same thing?
Yeah, I think so. Definitely figure 1, it gives you a nice overview of the process that they followed and why they were following it. So they give you some insights into how they build the network, which is really helpful for anyone that is planning on adapting the same procedure for other purposes. And also just provide a bit of information of the different databases that they used in order to build the network, as well as some of the traits that they were looking or how they categorized those traits, basically.
Yeah, and one of the interesting things that sort of struck me is, if you're looking at figure 1 and you sort of focus on panel A for a minute here. It's interesting because the network they came up with isn't actually that large in the context of like the kinds of networks that people can build these days.
Yeah.
So, you know, they had 18,000 nodes and about 570,000 edges, which is still a lot, but, you know, I've seen people describing networks where the node and edge count gets into like the millions almost. So I don't know, to me, it kind of strikes me in terms of like, it's not necessarily the size of the network you build, but more about like how it's constructed and then the ways in which you're able to actually analyze that data. Did you have a similar thought, or was that just me?
No, I think it makes sense. I think sometimes you just rather focus on the value of things. For instance, in I think it's panel C in figure 1 as well. They basically go over and say like how they focused first on those genes that had higher priority or were more relevant, and from there just assign scores to the genes that were very close to it, depending on how close to those genes the other genes were. And yeah, I think that just highlights the fact that it's more important to focus on things that you're interested in rather than gathering a lot of information about something that may drown the real conclusions that you want to get to.
Yeah, I think that's definitely true. You know, in today's era of big data, it's easy to come up with huge data sets, but often a lot harder to actually make sense of them, which at the end of the day is really why we generate the data in the first place. So it's nice to see studies like these where they really focus on the analysis part of it. Yeah, I think figure 1, the rest of figure 1, sort of panels D and E, we can kind of safely just skip over those for our purposes here. I think they're more to do with validation and stuff. But if we then go to figure 3, which I think is the other really key part of this, and really here, the big one, again, I think is panel A. So what they essentially did here was they made these sort of trait gene module associations, and then they clustered that together to come up with a list of sort of related traits and all of the different modules that are shared between at least 2 of those. And this is where they really get into that second kind of key thing from the paper, which is all about pleiotropy. So for listeners who may not know what that term is, or may just need a bit of a refresher, so pleiotropy is essentially when a gene or some other kind of genetic element has an effect on more than one trait. So, knock down this gene and you see a wide range of phenotypes, for example. And it was interesting because you can clearly see that there is some overlap between these very disparate traits and these gene modules that they had identified. What were your sort of thoughts about not just the figure panel, I guess, but also just this general idea and the finding that so many genes fit into this sort of pleiotropic module thing that they had come up with?
Yeah, I think that the one thing that I can highlight about figure 3A is the fact that they really brought in a lot of other information that wasn't just related to the GWAS studies. So for instance, they were looking at Campbell targets, Glyn-Barr variants, and also at information available at the mouse KD database. And I think that really just ties up everything together really nicely in the sense that it provides extra information that you would need in order to make these conclusions about like basically the purpose of the study, where they want to propose this method as something that can be used for drug repurposing, or to figure out if something is related to a specific side effect and so on. So by really tying together that information to how many hits you get, for instance, in clean barbarians, yeah, definitely you can get a lot more insight than by just looking at the data isolated.
Yeah, definitely. And not quite from figure 3. I think this was actually a point from a previous figure, but just because you bring up the idea of associated targets and other sorts of information, I think somewhere in this paper too, they talk about how some of these associations that they've identified, there are existing drugs that already target one or more of the genes in a module, but then there are other associated modules where there's just nothing that exists already. So also kind of interesting that spread of like some areas we've looked at before and we have therapeutics for, and then there's other areas that we know from things like genetic variant information, but we don't actually have any way of really getting at them just yet, it seems. So
yeah, for sure, it's definitely a great tool to uncover anything that you otherwise wouldn't be able to find with other methodologies.
So kind of a fun question here. So if someone just naively asked you, what would you think would be a pleiotropic gene in humans? What kind of gene would that be? What would its function be? What would your sort of gut response to that be?
Oh, that's an interesting question. And I think that's something that they do touch on this paper a little bit. Well, anything that would have to do with cell reproduction, like every single cell has to reproduce. So I imagine that the genes involved in that would certainly come up a lot in pleiotropic studies. And yeah, it's interesting that you bring that up because I believe that towards the end of the paper, the authors do talk about how you may see a lot of these type of modules or group of genes that are involved in a specific function, where you see something like extracellular structure or things that are very inherent to how everything functions in the body. And of course, there are a lot of risks on targeting a drug or having a drug that targets one of those mechanisms, because you don't want to mess up those very important and relevant mechanisms. But I think that there's still a lot of value in those pleiotropic traits that not necessarily fall into those highly important mechanisms, I'd say.
Yeah, I think I feel the exact same way. And what's interesting to me is that gut feeling of like, it's probably something more fundamental, that seems to really play out in the actual data that they come up with in this paper too. So, just as an example, if we stay on figure 3 for a second, if you look at panel B, they have, the modules that are the top overlappers between traits. And let's see, the top, what is this, 6, I think? So they have extracellular structure, regulation of transcription and proteasome, GPCR signaling, protein polyubiquitination, RNA processing, and endocytosis or clathrin-mediated endocytosis. So just really common sort of fundamental stuff. But it's always nice when, our expectation from our understanding of cellular biology and biochemistry actually does in fact play out when we look at the data. I think that's one of the nice things about science, is when you have those moments and everything kind of fits together nicely. So
exactly. Yeah, I totally agree with that.
And sort of the rest of figure 3 is really just them giving some examples of these shared modules and a little validation around examples from other parts of the literature where they can clearly point to like, yeah, we know this gene's actually involved in both of these types of traits. And I think that's just a nice validation exercise that they went through. Yeah, so I guess like those were, to me, those were the two big ones. But if we think about some of the other key insights that they found outside of sort of, you can build this network and use it to seed GWAS results, and pleiotropy is sort of rampant in these important modules. What were some of the other key things that you took away from the paper, did you think?
Yeah, one thing that I found interesting was the fact that several of the examples that they went through of how these these networks were basically, or could be used for different conditions, is the fact that there were several of those conditions fell within the immune realm, I'd say. So, for instance, like I think that they were talking about like immune diseases, cutaneous and respiratory immune diseases at some point, and I believe that some others were based, like were focused on IBD and so on. So I just found it very interesting that they were able to find a lot of pleiotropic traits in those type of conditions, and I just wonder if there's something linked to immunity, like between pleiotropy and immunity. And yeah, I just found it very interesting. Like it was something that they discussed a couple of times, and yeah, I just wonder if there's something else that might be interested in there.
Yeah, no, that's an excellent point. There's probably a lot to unpack there, none of which we can do in the short period of time we have today. But yeah, I guess to me, it probably just comes back to that idea of, the immune system, at least for multicellular organisms like humans, it's just so fundamental, and it has the potential to touch a lot of different cell types and a lot of different tissues. So dysregulation is probably bad in a lot of cases, I would say.
Yeah, exactly.
Yeah. I think the last thing that I wanted to say here is a little bit later on in the paper, and I don't really want to go too deeply into the details, but it was interesting too that they were able to use the same network that they had constructed to effectively prioritize a candidate gene set in IBD and come up with a couple of genes that they would consider to be maybe like, I don't want to say high risk, high reward, but ones that didn't have a lot already known about them, but from their network seemed to be good candidates for eventual drug targets. And the reason I bring that up is just because this idea of target identification or prioritization is just such a fundamental part of drug development. It's always nice to see new tools that can be used for those purposes.
Yeah, for sure. It's interesting how, so on that point, they were talking a little bit about how manual curation was able to, through manual curation, they were able to see that curated genes had higher network scores, in the sense that they were basically confirming that this network approach that they were following was really showing things that you would expect to come up as true in this type of study. And they also follow a different process with the locus to gene score that is available at open targets. And yeah, and so basically similar results with that, but it's just interesting to see that you need to take these extra steps in order to get the targets that you want to focus first, the genes that you want to focus on first, because when you have a lot of data, you can't just go through the 1,000 hits that you may get in a study. So you have to start somewhere. And it's interesting that, and it's very relevant as well, that they really introduced and put a lot of value into this gene prioritization concept.
Okay, so I think, we sort of covered the key findings of the paper now, I think, quite nicely. And of course, people who go and read it afterwards will likely be able to dive into all of the details that we couldn't really get to today. But one of the natural questions that always comes up after reading a paper, I think, is, what other similar work is being done in these areas, or how could this work be expanded into the future? So I just wanted to sort of briefly touch on that. So did you have any thoughts around that particular area?
Well, I think that I haven't come across with a paper that is just like this one. So the first thing that I would be interested in seeing is how many people are going to start adapting this type of methodology to other purposes, and see, for instance, if here they focused on pleiotropy, but I would be very interested to see what other things they may be able to draw conclusions on that are a little bit different, still based on these network approaches that use GWAS data as a starting point.
Mm-hmm. Yeah, definitely. I think the only thing I'll add is just, it's kind of funny because, I feel like lately there's been a real hype cycle in terms of, all these new AI approaches, and there's sort of been a real explosion in AI. But, then the most recent conference I went to, you started seeing lots of people talking again about knowledge graphs and graph-based approaches, and even how to combine that with things like large language models. So just seems like these fundamental things around like using data structures and building a good data structure to power your eventual analysis, whether it's something that's more traditional or whether you're using some kind of machine learning. It's just, there's always got to be that focus on quality data and like a well-designed system at the base of all of it. So
yeah, definitely. Would be interesting to see how they will combine this type of information together.
Okay, so the next thing that that we should probably quickly talk about is, how this can be useful for people who make use of DrugBank. So for anyone who's unfamiliar with DrugBank, we have this large platform of drug and related biomedical information that we ourselves do a lot of authoring of content, but we also pull in information from other sources, use things like artificial intelligence to help power a lot of the curation that we do. And this information is available through DrugBank Online, which is our free to use website, or we have also options for academic and commercial users. But all that is to say is, there's a lot of information that we have, and it's always interesting to think about how DrugBank could fit into studies just like these ones that our users might want to try to come up with. So what are your thoughts on that? How do you think people could actually leverage a resource like DrugBank to do research like what we saw in this paper?
Yeah, I mean, I think that the targets data set that we have would be greatly valuable in this case. And we can see that in the paper, they really focused on not just sourcing a single database, like they really did their due diligence and look through different databases that may be helpful in order to draw the conclusions that they got to. But yeah, so the more that you focus on integrating these type of databases that provide robust information, I think that the higher the probability that you would be able to get a result that makes sense, which is what they did in this paper.
Yeah, I think that's definitely true. There's also potentially things that you could add in here too, like for any of those drugs that they were talking about in terms of known drug targets or indications or something like that. That kind of data can easily come from DrugBank, and then you could potentially expand a network such as this one to include the actual drugs directly within it, and maybe even try to predict new connections between existing drugs and some of these well-connected nodes using various algorithms that already exist. So,
yeah, and another thing that may be interesting would be to also incorporate data sets that we have, for instance, in, as you mentioned, like indications and also adverse effects that perhaps could complement this type of analysis by going further than just taking a look at the targets and being able to connect to other targets just through that information.
okay, so we're coming towards the end of our time now, and I think it's only fair to turn our attention back to the paper itself for the final section here. And this is all about summarizing big takeaways and potential future directions that you could see for this type of research. So I'll let, if you want to kick us off here.
Yeah, for sure. So one of the things that I did notice, because I have a background in basic science, which is very different from these large studies that include lots and lots of data. And I think that compared to more traditional approaches, like the ones that people were following even before GWAS studies were performed, is that by using these large data sets and analyzing the data, it just takes the guessing out of research, in the sense that, for instance, yes, you may be able to assume that something that is that can be used to treat, let's say, cutaneous immune diseases. Maybe you could look into rheumatoid diseases or drugs used to treat rheumatoid diseases in order to follow some drug repurposing approach. However, you would be guessing at that point. You're making an assumption. You don't really have the data to back that type of decision. And I think that following this type of approach, it just makes the workflow for scientists working in drug repurposing a lot more efficient, in the sense that you don't have to be doing trial and error anymore. You can just follow a very, very thorough process that gives you the higher probability targets that you should be focusing on, and focus on the important things first, basically.
Yeah, that's definitely an excellent point. That's kind of the classic curse of the bench scientist, where they have a hunch, but tracking down all of the data that you would need to really make that hypothesis can take so much time if you're just doing it the very traditional manner of reading papers and looking within the literature itself. So it's nice to have these sort of larger scale approaches that are a little bit more automated, a little bit less biased than some other more traditional approaches we might have. So I think for myself, there were sort of three big things that I took away from this. So the first one was that these GWAS, or genome-wide association studies, they continue to be a really interesting source of data for understanding gene disease associations in what is a relatively unbiased way, although there's always going to be bias in these studies, like historically in terms of patient population selection and those sorts of things. But that, there's still a lot more work that needs to be done to really fully understand and interpret the results that we get from those studies. It's not like a one and done thing where we can just jump straight from significant association to great, we have a drug target now.
Yeah, definitely. Oh yeah.
Sorry. Yeah.
No, I was just gonna add on to that there are certainly a lot of limitations to GWAS studies that people are aware of, but I think it's important to remember that the data that you get from a GWAS study is just as important. Regardless of these limitations, it just depends on how you work with that data and what are you going to do afterwards with it to really get value out of it.
Yeah, 100%. Fully agree. And on that note, actually, sort of my second takeaway was specifically around this idea that, I've seen graphs used in a lot of different ways, and people will put different kinds of information into their own knowledge graph. But in this study particularly, they just show that even a relatively simple graph can basically turbocharge your ability to interpret the signals that are present in GWAS data. And I just think it's nice when a really clear and well understood data structure can help you to do that. And it's not like you have to develop a bunch of new methods. It's all in the preparation and analysis of something that's already pretty well established.
Yeah, definitely.
Yeah, and then I guess the last thing that I sort of took out of this was that it was interesting in that there was a lot of congruence between things like the GWAS data and variants from ClinVar, for example. Then there was also a lot of differences between the GWAS and the ClinVar and things like mouse, knockdown phenotypes or yeast knockout screens in terms of, associated genes and even pleiotropy. So it's just interesting. I think there's still more work that we need to do to really fully understand all of this data that we're collecting. But it is nice to see at least some agreement between very different types of data and data collection methods.
Yeah, for sure. And I think that one of the things I just want to jump into that point that you were discussing about the knockout phenotypic studies. So that's one of the limitations that the study does talk about how, yes, you are unable to look at a lot of cell-to-cell relationships when it comes to knockout phenotypic studies compared to the approach that they followed here that really nicely highlighted that type of information. However, they do say that because of the approach that we're following, it just comes back to that point that we were discussing before, where a lot of these highly relevant mechanisms that you can't really mess up with when you're trying to figure out which target you want to focus on for drug repurposing or for other drug development purposes. But I think that still it's still very valuable in the sense that you probably want to put together all your cards on the table and see how everything looks, rather than follow just a single method and go with that approach and not compare it to other things that you would be able to have in your hands in order to make the right decision.
Yeah, 100%. Okay, so that's it for today's session. Thanks so much for joining us on Peers Reviewing. If you're looking for more, feel free to subscribe and stay up to date on all of our latest episodes. You can look at go.drugbank.com, start searching our knowledge base, and to keep up to date with all of the information that's present there. Of course, you can see us on social too. We have a LinkedIn. And if you have ideas for future papers you'd like to see discussed on Peers Reviewing, please send them our way. And happy sciencing.